Hi everyone, I'm Shamsuddin Bhuiyan from the Pavlidis Lab at the University of British Columbia here in Vancouver, Canada. Thank you everyone for joining me today to talk about my project, prioritizing genes with functionally distinct splice isoforms. Alternative splicing is a post-transcriptional modification that may increase a gene's functional diversity. Classic textbook example of this is gene calc-A, where in neuronal cells it expresses a splice variant called CGRP, which is necessary for vasodilation. On the other hand, in thyroid cells, it expresses a splice variant called calcitonin, which is necessary for calcium phosphate metabolism. Because calc-A is a gene with two or more necessary splice variants, we define this as a gene with functionally distinct splice isoforms, or FDSIs. Now, calc-A is a great example of a gene with FDSIs, but we don't know very many cases like it. Even though 95% of the mammalian genome undergoes alternative splicing, the functional consequences of most remains unknown. Looking further into the literature, it seems unlikely that many genes would have FDSIs. mRNA splicing is a process full of stochastic elements. The splicing machinery is constantly present in the cell and is constantly bombarding mRNA molecules. Introns are mutational hotspots that easily give rise to splice site recognition motifs. Because there's so many avenues of randomness, the question becomes how much of alternative splicing comes from these stochastic elements versus how much comes from a biologically necessary regulated process. Now, given that most splice variants are not likely to evolve under selection, given that most splice variants are lowly expressed, and given that most splice variants lack protein-level evidence, many members of the field have concluded that it is unlikely that most splice variants are functional, and thus it is unlikely that many genes have functionally distinct splice isoforms. However, some genes must have FDSIs, no matter how few. So we were wondering whether it's 5% or 40% of the genome, what are the genes with functionally distinct splice isoforms? The idea behind our approach is to prioritize genes with multiple desirable splice variants, such that we're finding the most likely candidate genes with FDSIs, the low-hanging fruit, so to speak. Now, by properties of a desirable splice variant, I mean properties that make the splice variant more likely to be biologically necessary. So, for example, if a splice variant is conserved, then we think it's more likely to be functional than if it isn't conserved. And similarly, if a variant is not well expressed, then it's less interesting than a splice variant that is expressed. Furthermore, if a splice variant encodes for an open reading frame that encodes for a functional protein domain, we think that's far more interesting than a splice variant that doesn't encode for an open reading frame or encodes for an open reading frame without an annotatable protein domain. Now, you may agree or disagree with these criteria or thresholds, but we think they are a reasonable way to prioritize the most interesting potential cases of genes with FDSIs for further study, especially in light of what we've learned about all the noise in the alternative splicing process. So my prioritization pipeline uses a combination of already available tools as well as tools that I wrote. At the initial step, I take raw long-read data, process and cluster them into genes with multiple splice variants. Because long-read data can be erroneous at specific splice junctions, I use corresponding short-read data to alleviate for these errors. So my mouse brain and mouse transcriptome data sets provide me with just over 16 million reads, which gets processed and clustered into 6,799 genes with 41,281 splice variants. Interestingly, about 95% of these splice variants are not present in Ensemble, making them novel to our computational databases and computational tools. I then annotated each splice variant using the functional genomics that we talked about earlier. So each splice variant was annotated with protein domains from the conserved domain database. Each splice variant was annotated with a conservation score. These were calculated in a number of different ways, such as FASTCons and PhyloP, which is based on multiple sequence alignments, and homology-based scores like BLAST. And finally, my annotations for appreciable expression and coding potential of the splice variants were based on scripts that I wrote. So you'll also notice that underneath each functional genomics category, there is a percentage, and this percentage represents the proportion of genes that passes the functional genomics category. For example, 83% of the 6,799 genes had 2 splice variants that each encoded for a protein domain. On the other hand, only 5% of the 6,799 genes had at least 2 conserved splice variants. And if a gene had multiple splice variants that passed all 4 of these boxes, we considered it a gene likely to have functionally distinct splice isoforms. And so out of 6,799 genes, we prioritized a list of 72 genes likely to have FDSIs. Now, because our data has such low depth and we don't have very many tissue types, this is probably not a comprehensive list of genes with FDSIs, but we do believe there is some promise. So moving forward, the key finding from my study is that of the 6,799 genes, we prioritized a set of 72 genes likely to have FDSIs. And that's 1% of the genes with multiple splice variants in this data set. I didn't get to talk much about this, but our preliminary results support trends from short read data. Primarily, most genes have one dominantly expressed splice variant, and few genes have multiple conserved splice variants. And these results are preliminary, mostly because we lack sufficient sequencing depth or multiple different tissue types. But hopefully, with more long read data, we can strengthen these claims. We're also looking to apply this approach to other contexts, such as looking at targeted transcriptomics of genes with complex transcript structures like CACNA1E and the splice variants of CACNA1E. And finally, our experimental collaborators are looking to validate some of our interesting genes, such as GSTZ1. With that, I'd like to thank my lab for their support, my PI, my committee members and collaborators, as well as our funding sources. Thank you very much.